Welcome to this month's April 20th, 2024 Ask Your Herb Doctor, Ask Your Herb Doctor, KMUD Galberville 91.1 FM. Oh, it's the 19th. Oops, it's the 19th of April. Sorry, folks. Anybody out there recording us chronologically across the wide web and posting these things all over the place without permission, please don't. And it's the 19th of April. Okay, so tonight's show, sorry we got off to a bit of a late start, had a little bit of live streaming issues here, but that's all sorted. Tonight's program is going to be on berberine as a specific compound, and then biofilms. Biofilms are really quite a new discovery. I mean, since 2016 maybe, but it's getting mainstream traction at this point. So we're going to talk about berberine and then biofilms and discuss some of the implications of biofilms and the resistance to antibiotics and treatments, and why some people with certain conditions, of which they're finding now the biofilm is the reason for the stubbornness or resistance to treatment, is why they're not getting better. So you're listening to Ask Your Herb Doctor, KMUD Galberville 91.1 FM. From now until 8 o'clock, we will be live streaming the program. So my name's Andrew Murray.
My name's Sarah Johannesson Murray. Welcome to tonight's show.
We're both licensed medical herbalists. We consult with clients about a wide range of conditions. And our website is westernbotanicalmedicine.com. You can also visit our site to see a list of our organic medicinal herb tinctures, and our contact details are there also. Alternatively, we can be reached on our toll-free number 1-888-WBM-HERB, and our Western Botanical Medicine Twitter's account is at WBM-HERB. Yeah, and our YouTube channel is Western Botanical Medicine. So links to both our Twitter and YouTube channel are also on our website at westernbotanicalmedicine.com. So to continue, so we weren't live streaming in March, but we're back now and want to continue. It's kind of broader topic, really, of things related to resistant to treatment pathologies, of which we've gone through things like SIBO and SIFO in the last few months' programs. That's certainly part and parcel where some treatment is very difficult for some patients that either don't get remission, or they get a very brief remission followed by a relapse, or they don't get a remission with or without conventional medicines and herbal medicines. So whether they're taking rifaximin and neomycin because they're going the traditional medical route, or they're using alternative treatments, some people just don't get remission, and some people get a brief remission followed by relapse, which is fairly common. And some people take 2 or 3 rounds of treatment, whether alternatively or conventional, to try and eradicate the particular archaea that they're dealing with, whether it's hydrogen dominant or methane dominant. Anyway, without going back into that too much, just wanted to start the show talking a little bit about the compound berberine. So berberine is typically used in the treatment of things like hypercholesterolemia, so high blood cholesterol, menstrual irregularities, of which there's a hormonal component, and PCOS, polycystic ovarian syndrome, obesity, and diabetes, just to name a few syndromes or metabolic defects that people have that can be treated using berberine through its action. Now, berberine, as I said, is what they call a quaternary ammonium salt. So if you look at the structure of ammonia, the NH4 group, the nitrogen atom is joined by 4 products. 4 arms, 4 branches are joined to the ammonia group, forming what they call the quaternary. So you have tertiary and quaternary ammonium salts. So berberine itself, it comes from a quaternary ammonium salt from the proto-berberine group of benzyl isoquinoline alkaloids. These occurring naturally as secondary metabolites in some plants, including species of Berberis, from which its name is derived. It's also found in other medicinal plants from Coptis chinensis, the Chinese golden thread, through Mahonia aquifolium, the Oregon grape, Hydrastis canadensis, golden seal, through Eschscholzia californica, the California poppy. Now, quaternary ammonium compounds are lethal to a wide variety of organisms except endospores and non-enveloped viruses. So we'll cover these in a bit and give you a bit of a background lesson, if you like, a kind of quick 101 of microbiology from a bacterial perspective as well as a viral perspective, just to illustrate how these compounds and how compounds that are used in the treatment of disease generally are targeted at specific organisms and how they work. So it's possible to solve the endospore problem by adding chemicals that force them to germinate. So endospores are essentially like a hibernating form of an organism here, and then in general, these things, these compounds, these kind of quaternary compounds, have a reduced efficacy against gram-negative bacteria, mycobacteria, and bacteria in biofilms due to them having additional layers that need to be penetrated or disrupted. And some bacteria, such as MRSA, the methicillin-resistant Staph aureus, have acquired resistant genes also that pump this cation out of the cell. So bacteria are always developing strategies by which they can overcome that which is forcing them to die or causing them to die. And so they elaborate very many different strategies, and actively pumping components out of cells so that they don't build up to toxic levels is one of those strategies. And I'm not saying that I believe in membrane pumps, folks, because I think that's been well discredited, very well and very succinctly. But the mainstream science and teaching still continues to perpetuate the membrane pump theory. Anyway, so endospores then. Endospore formation is usually triggered by lack of nutrients and usually occurs in gram-positive bacteria. Now, endospores enable bacteria to lie dormant for extended periods, even centuries. And there are many reports of spores remaining viable for thousands of years. Okay, examples, and this is where it's going to get a little microbiology 101, and it's not exactly herbs and herbal medicines, but I just want to give people the kind of relationship between these particular bacteria and some of the treatment strategies used to overcome their innate kind of ability to resist the killing effect of either the cell or chemicals that are used in the form of antibiotics, etc. So just to give you some examples of the bacterial species that can form endospores, they're mainly in the genus Bacillus. So the Bacillus cereus, Bacillus anthracis, Bacillus thuringiensis, and then Clostridium species, two of which are Clostridium botulinum and Clostridium tetani. Now, people hearing these Latin name descriptors for these particular bacteria will recognize them, and we'll go through them. So the endospore, then, the kind of latent dormant form, consists of the bacterium's DNA, ribosomes, which transcribe it, and then large amounts of dipicolinic acid. Now, dipicolinic acid is a spore-specific chemical that appears to help in the ability for endospores to maintain dormancy, and the chemical accounts for up to 10% of the spore's dry weight. So getting into these brief description about these particular bacteria and what they do, and then we're going to kind of wrap it up into a collective thought here, where these are going to be tied to their treatment and show you where the resistance in treatment occurs, that these bacteria can certainly be part of a biofilm, and how that biofilm can be overcome with the use of different components. So Bacillus cereus. Now, these bacteria are both spore forming and biofilm forming, presenting a large challenge to the food industry due to their contamination capability. So biofilms of Bacillus cereus most commonly form on air-liquid interfaces or on hard surfaces such as glass, and Bacillus cereus display flagellar motility, so they can move around, which has been shown to aid in biofilm formation via an increased ability to reach surfaces suitable for biofilm formation, to spread the biofilm over a larger surface area, and to recruit planktonic or single free-living bacteria. As we go through and we discuss biofilms, we'll discover that they are a fairly complex collection of different bacteria, substances, and extracellular matrix. So the second bacillus, or second species of bacillus, which again can form endospores here, is Bacillus anthracis. So this is another gram-positive rod-shaped bacterium that causes anthrax. So people probably recognize anthrax from the kind of, doomsday type bioterrorism kind of modality in which people are, certainly scared into paying ransoms for whatever it is, or, presidents are sent a letter containing anthrax spores. So it's a deadly disease to livestock principally, and occasionally to humans. It's the only permanent obligate pathogen within the genus Bacillus, and its infection is a type of zoonosis, so it's a kind of animal-human jumping the species barrier transmission, as it's transmitted from animals to humans. So it was discovered by the German physician Robert Koch in 1876, and it became the first bacterium to be experimentally shown as a pathogen. This discovery was also the first scientific evidence for the germ theory of diseases. So the infection can be treated with common antibiotics such as penicillins, quinolones, and tetracyclines. So it's a very treatable situation, and when we were studying herbal medicine and we had pathology as one of our weekly lectures throughout year 2, we had microbiologists that were lecturing, and anthrax was relatively, well, I say common, but you always had X amount of cases of anthrax in England, and typically from the hide of an animal. So, anthrax-bearing animal carcasses that were either came into contact with humans because of trapping, or cows that contracted anthrax, and the hide of the cow was being hauled around by either the slaughterer or somebody else working on the farm of disposing of an animal. So it was not uncommon, but just to say that anthrax itself is relatively easy to treat if you can recognize that what you're looking at in the eschar, which is a common presenting kind of skin manifestation of it, not the pulmonary form, but the skin form, a cutaneous form, it can be quite easily treated with antibiotics. So, as I said, it's one of the few bacteria known to synthesize a weakly immunogenic and antiphagocytic protein capsule called the poly-D-gamma-glutamic acid capsule, and that distinguishes, or rather disguises, the vegetative bacterium from the host immune system. So this particular poly-D-gamma-glutamic acid is a kind of counterfeit and a kind of a cloak that the bacterium hide behind, that the immune system doesn't recognize as foreign, and so it's not actually not able to mount an immune response to it. So most bacteria are surrounded by a polysaccharide capsule rather than this poly-D-glutamic acid capsule, which provides an evolutionary advantage to Bacillus anthracis. So polysaccharides, then, these kind of sugar components, are associated with adhesion of neutrophil secreted defensins that inactivate and degrade the bacteria. So neutrophils, the arm of the immune system that we have, ultimately produce compounds called defensins, and they inactivate and degrade most bacteria, but they won't get to this particular capsule, the poly-D-glutamic acid capsule, because it's not recognized as something that the neutrophil would mount any kind of response to. So by not containing this macrocapsule in the capsule, Bacillus anthracis can evade a neutrophilic attack and continue to propagate infection, so hiding from the host. The difference in the capsule composition is also significant because poly-D-glutamic acid has been hypothesized to create a negative charge, which protects the vegetative phase of the bacteria from phagocytosis by macrophages. So two different methods of evading the immune system. So like Bordetella pertussis, the causative organism in whooping cough, it forms what they call a calmodulin-dependent adenylate cyclase exotoxin, which is a virulence factor known as the anthrax edema factor, along with anthrax lethal factor. So both of these can cause a fairly rapid death if it's untreated, especially from pulmonary forms of it. It bears close genotypic and phenotypic resemblance to Bacillus cereus and Bacillus thuringiensis, which is the third of the bacilli that we're going to just briefly talk about. Now,
So, sorry to interrupt you there, but are these bacilli hiding from the immune system in these biofilms?
No, only anthrax. I'm just saying that anthrax produces, it has a poly-G glutamic acid capsule, which its kind of DNA is housed in. So therefore, it evades the immune system because neutrophils don't recognize this.
So that is a form of a biofilm. So that's why so many people can die from anthrax.
It's not a biofilm, no. It's just saying because this particular bacterium forms this component around its genetic makeup, that component is not recognized like most bacteria surrounding themselves with polysaccharides as a capsule are recognized. So it's not like a sugar that's around the bacteria like they do mostly.
So it evades the immune system. Anthrax is so deadly because it evades the immune system in the same way that like a biofilm will hold another type of bacteria, and the immune system won't mount a response.
Or won't see it. Yeah, won't see it because it's hiding within a biofilm. That's the same way that Bacillus anthracis would cause a lethality regularly if it's untreated because there's no real host immune defense for it.
So the only hope you have to treat it.
Well, it's if you recognize it, number one. If you recognize the symptoms of it and the signs of it, then yes, you need antibiotic treatment.
Right.
Yeah. So Bacillus thuringiensis, though, for those people perhaps are horticulturalists and are interested in biological control, it's again Bacillus thuringiensis, or BT as it's known. It's a gram-positive soil-dwelling bacteria, most commonly used. It's the most commonly used biological pesticide worldwide. It occurs naturally in the gut of caterpillars and the caterpillars of various types of moths and butterflies, as well on leaf surfaces, aquatic environments, animal feces, insect-rich environments, and flour mills and grain storage facilities. So it's also been observed to parasitize some moths, such as the dried fruit moth. In laboratory experiments working with this species, many of the moths were diseased due to this parasite. And it's first discovered in silkworms in 1902. Now, during sporulation, many BT strains produce crystal proteins called delta endotoxins that have insecticidal action. So this has led to their use as insecticides, and more recently to genetically modified crops using BT genes, such as BT corn. Many crystal-producing BT strains, though, do not have insecticidal properties. The subspecies israelensis is commonly used for control of mosquitoes and of fungus gnats. So the last two bacteria that I wanted to talk about here in relation to biofilm and how biofilm can be responsible for the persistence of a particular pathology that's difficult to treat, the two Clostridiums. So if people are, you probably heard of this Clostridium botulinum, where you recognize that right away from the specific epithet botulinum, causing botulism. And then the last one, Clostridium tetani, so causing tetanus. So just to deal with Clostridium botulinum first, that's another gram-positive anaerobic spore-forming motile bacterium with the ability to produce botulinum toxin, which is a neurotoxin. It's responsible for foodborne botulism, and this is typically because of the ingestion of the preformed toxin, as well as infant botulism, which is an intestinal infection with toxin-forming Clostridium botulinum, and then wound botulism, infection of a wound with Clostridium botulinum. It's heat resistant. The endospores typically are quite heat resistant, are commonly found in the soil, and are able to survive under fairly adverse conditions. A lot of these forms are very resistant. So the spore-forming forms are typically like time capsules. Like I said at the beginning of the show, some have been shown to be several thousand years old, and they can just sit still and wait for ideal conditions to arrive again, and then just spontaneously germinate and become the virulent form. So Clostridium, as well as these other bacilli, are able to form endospores. So the Clostridium tetani then is a common soil bacterium and the causative agent of tetanus. And Clostridium tetani spores are extremely hardy and can be found globally in the soil or in the GI tract of animals. If inoculated into a wound, it can grow and produce a potent toxin, which is tetanospasmin, which interferes with motor neurons, causing tetanus. So you're listening to Ask Your Herb Dr. KMD Garberville 91.1 FM from 7:30 to the end of the show. You're invited to call in. The number, if you live in the area, 923-3911. And we'll be taking calls from 7:30 to 8. So I just wanted to briefly talk about enveloped versus non-enveloped viruses, and then we'll get onto the whole biofilm and the whole treatment that can be had. So the envelope versus non-envelope virus debate, then, the lipid bilayer membrane on the outer part of the virus differentiates enveloped versus non-enveloped viruses. Viruses are composed of two main components, then the viral genome, which can be RNA or DNA, and the virus-coated protein capsid that surrounds the genome. If the virus particle contains only these two elements, it's called non-enveloped. But if the virus particle contains an extra lipid bilayer membrane surrounding the protein capsid, it's called an enveloped virus. And non-enveloped viruses, also known as naked viruses, are typically more virulent than enveloped viruses because they usually cause host cell lysis. So when they replicate within the cell, when they emerge from that replication phase, they typically rupture the cell. Okay. So this is one of the major differences between non-enveloped and enveloped viruses. So since non-enveloped viruses don't have that extra lipid membrane, cell lysis is the most common exit mode from the host cell. During this event, the viruses violate the integrity of the cell membrane, cause cell death and significant tissue damage to the organism, whereas non-enveloped viruses are more resistant to extreme pH, heat, dryness, and simple disinfectants. Some examples of non-enveloped viruses are noroviruses, enteroviruses, adenoviruses, and rhinoviruses. On the other hand, enveloped viruses are typically less virulent than non-enveloped viruses, and that's because they don't always cause cell lysis during their exit from the cell, although cell death often follows as a consequence of virus replication. Since enveloped viruses contain an outer membrane that surrounds the capsid during the viral assembly and exit from host cells, these viruses can use the host cell membrane itself to assemble their membrane, known as the envelope. This process avoids cell lysis and helps enveloped viruses escape the host immune system. Now, enveloped viruses also tend to be more sensitive to extreme pH, heat, dryness, and simple disinfectants. Examples of these enveloped viruses include influenza, human cytomegalovirus, HIV, respiratory syncytial virus, vaccinia virus, and human coronaviruses, SARS-CoV-2 being one of them. So that's the essential bacilli and the difference between the envelopes and non-enveloped viruses. But I wanted to get into the subject of biofilms. So why is the subject of biofilms becoming so relevant to human disease research now especially? So a biofilm, what is a biofilm? It's what they call a syntrophic community of microorganisms in which cells stick to each other and often also to a surface. These adherent cells become embedded within a slimy extracellular matrix that is composed of extracellular polymeric substances. Bacterial biofilms can be considered to be an emergent form of bacterial life in which communal life is completely different from bacteria that live as free-living cells. So biofilms are the predominant form of microbial life in the natural environment, and although they are the leading cause of chronic infections, they're equally deeply connected to our ability to bioremediate waste and toxic materials. The term biofilm was introduced in the early 1970s by microbiologist J. William Costerton and colleagues, who were studying the bacterial communities that form on surfaces in aquatic environments. They found that these communities were not simply random aggregates of individual cells, but instead were highly organized structures with specialized adaptive functions such as nutrient uptake, waste removal, and protection from environmental stressors. Biofilms exhibit emergent properties that arise from the interactions between the microorganisms that make up the community, as well as between the microorganisms and their environment. These emergent properties provide benefits to the microbial community beyond the benefit to any individual cell. Okay, so it looks like we've got a caller. We'll take this caller. Caller, you're on the air. Where are you from? What's your question? Uh-oh.
Oops, maybe that was my fault. Please call back, caller.
Yeah. Okay, the number is 923-3911 from now until 8 o'clock.
Are you going to get to nattokinase at some point?
Don't think I've got anything for nattokinase today.
I was going to ask you about nattokinase and lumbrokinase in treatment of biofilms, which is something I learned about in Lyme's disease.
Yep.
Yes, I was just speaking to a colleague about lumbrokinase today, who was suggesting that it helps increase circulation even when you use topically, and it's a natural form of Botox to help bring circulation to the skin surface. But I know it's also used in biofilm.
And somebody's calling back. Maybe it's that same caller. Let's just go right to them. Caller, you're on the air.
Where are you from? What's your question?
Hi. It was me. I got cut off somehow, but I know you're having trouble, so yeah, it's fine. Here I am.
What's your question? Hello?
Hello?
Hi, yeah, you're on the air. What's your question?
My question is, I'm asking, I know you're talking about biofilm, and I know I have a candida condition. Does candida as yeast form biofilm as well?
Was that the question?
Yeah, so I think the caller's asking if candida, the yeast organism that can commonly inhabit people's mouths.
God, you're so scratchy.
Oh, you can't hear me?
Well, you're very scratchy, but I think I can hear you. And can you repeat what you asked me? I'm sorry.
Okay. So I was just repeating your question to Andrew. I think I heard you correctly. It's a little hard to hear you as well. But I think your question was, is candida, the yeast organism that can inhabit the bowels and the mouth and the sinuses, does that also form biofilm?
Yeah.
To evade the immune system?
Yeah, the simple answer to that is yes. So a little bit later on in the work that I prepared for the show, we go over those some of those more common organisms for which biofilms are completely the reason why people don't get over their whatever drug they're taking or herb they're taking, even for the treatment of that organism.
Yeah, I'm taking Oregon grape and golden seal sometimes, and turkey tail and conch for the other overlying issues. But it's good to know that candida has a biofilm issue, and I just have to modify my lifestyle more. It's really hard.
Well, I was going to, towards the end of what I'd arranged...
Sorry, say it again.
I was going to discuss later on in the show particular compounds which are well known to reduce the biofilm, to make those things like the berberine in golden seal or the other herb that you mentioned, which is a well-known antifungal, to make those effective, because they are effective directly. It's just when the organism that you're trying to target with that particular compound is hiding within a biofilm, the biofilm itself the biofilm itself prevents that compound from reaching the organism. That's why they've developed this strategy. And the Candida doesn't have to necessarily create a biofilm. The reason why some people create biofilms and not is another debatable subject. And
That's probably genetic, right?
well, I'm sure there's environmental
Possibly, partly.
I'm sure there's environmental stressors within each
Oh,
person
sure, yeah.
that make
We're up
them
against, man.
that make them individually more susceptible
Yeah.
or not.
And I just want to say really quickly is that the newest Consumer Reports, which is a very credible organization, has come out against fragrance, has come out against all these fragrances.
The fragrances.
That was really good. So, okay, I'll
Good.
listen to the answer off the air. Thank you so much for your show. Thank you. You guys are wonderful. Bye.
Thank you for your
very
call.
Welcome. Thanks for listening.
And I just wanted to mention, because she mentioned she uses golden seal and Oregon grapefruit,
Right, they both contain-
another herb that contains a lot of berberine, which is the active compound in there, is Berberis vulgaris, which its common name is called barberry. And we have all three of those tinctures. We have golden seal, and we have Oregon grapefruit, as well as barberry, and they all contain berberine. And
they grow here excellently. So if you have any land at all, having a lot of Oregon grape is not only good for the wildlife because it's a native, if you get a native variety or transplant some native, but you're also, your backyard pharmacopoeia is really enhanced.
And
it's a nice
beautiful.
plant. Yeah,
Yeah.
you can make jelly. The birds love it.
yeah, we have several different ornamental varieties of barberry in our garden, and we also have the ornamental Oregon grapefruit. But even the native Oregon grapefruit is really beautiful, and they love it here. They're very pokey plants,
And
but they're really beautiful.
I suspect the native ones might be more medicinal just because I have those biases and they're growing
Although
more wild and right where
Yeah.
they want to be living.
when you dig up the ornamental ones, though, when we've transplanted some ornamental barberries, their roots are still bright orange.
And if you're digging it up and making your medicine out of whatever medicine it is, if you're actually digging it up from your own environment, I think that in and of itself is going to make your medicine a lot stronger.
That's part of the healing process in and of itself.
Okay.
But what other thing I wanted to mention about golden seal is that we are growing some golden seal too. It's not native here to the west coast of California, but it has hydrastine, which it also has very similar properties to berberine, which all three of those plants contain berberine. So that's a little botanical side note there, Andrew.
Thank you for that. Okay, you're listening to Ask You, Dr. KMD Garberville 91.1 FM. From now until the end of the show, 8 o'clock, you're invited to call in with questions. We're talking about biofilms. We discussed the quaternary ammonium compound berberine, and going to kind of interject that with the treatment for different pathologies of which we've yet to discover these and outline them for the treatment of breaking down biofilms so that these compounds can be as antimicrobial as we read they are. So in relation to disease processes then, biofilms can exhibit increased resistance to classical antibiotics and cause disease through device-related and non-device tissue-associated infections, posing a severe threat to health. So the device-related, we're talking about medical procedures here, and not to bash the medical people, but just to say that in the operating theater, sure, they go through fairly rigorous sterilization of tools and equipment, because I'm sure they're well aware of how resistant some of these bacteria are to classic antibiotics, organismic compounds, whether it's phenols, whether it's other potent caustic agents or alkalis or acids, as well as heat in the kind of autoclaving process. Some of these biofilms that form on operating procedure instruments are extremely resistant to a lot of the classical techniques for sterilization, and it's has been well documented, and that's how there's such a hoo-ha at this point in time about biofilms. There's such a hoo-ha about kind of surgery acquired infection, how there is such a huge case list of people getting resistant infections from surgeries, because different operating tools they're using can be resistant to the treatment that they're using to try and sterilize them, as well as other surfaces that, I mean, glass even, they talk about these biofilms maintaining themselves on glass for indefinite amounts of time.
Even through autoclave?
Well, they have certain temperatures for certain times. They say that is definitely curative, but and they would typically autoclave all the surgical instruments for sure. But how does somebody get a resistant bacteria as an infection from a hip transplant or from a urethral catheterization if it wasn't there to begin with? I mean, it's not like it's introduced from the person's own flora. It's actually acquired in the hospital, so it's definitely well recognized as a problem.
Well, that's how the MRSA
Right.
gets spread, is through biofilm.
Right. So in terms of the different things that these organisms do and how they can disrupt, general health. So talking about 5 different points here. So adhesion and structure. The biofilms form a variety of surfaces, both biotic and abiotic, and they create stable and robust attachments, making removal difficult and hence enhancing survival. Mature biofilms are 3D space-occupying structures with complex architectures. This causes spatial gradients and distinctive microenvironments that can vary in nutrient availability, oxygen concentration, and pH. So, from a division of labor perspective, the formation of different microenvironments can lead to the emergence of different subpopulations of genetically identical microorganisms within the biofilm, each exhibiting different behaviors and metabolic activities. Thus, the community can split tasks for maximum efficiency and cooperativity. However, inherent in this process is a need to protect the community from non-producing cells that, for example, can escape the metabolic burden of making shared goods but benefit from their production, leading to population collapse. So the division of labor is a different factor. 2, nutrient cycling. Within the matrix, biofilms can cycle and recycle nutrients, which can support the growth and survival of the microorganisms in the community. This nutrient cycling is facilitated by enzymes that are produced by the microorganisms in the biofilm, as well as physical and chemical interactions between the cells and the matrix. The process of nutrient cycling can occur both within a single isogenic community or between species. Then there's the enhanced robustness. Residents within biofilms are often more tolerant to environmental perturbations than planktonic or free-living microorganisms due to the protective properties of the biofilm matrix and the ability of the microorganisms to adapt to changing environmental conditions. Microbes in biofilm communities often demonstrate decreased growth and increased antibiotic tolerance, pH, temperature, and stress resistance, and immune evasiveness. Then there's the exchange of genetic material. Biofilms are known hotspots for horizontal gene transfer, which allows the exchange of genetic material between different microorganisms of the same or different species. This exchange can lead to the rapid acquisition of new traits that allow the biofilm community to adapt to changing environmental conditions, including the enhanced development of antimicrobial resistance. Then there is the collective action. Many microorganisms in biofilms use chemical signaling called quorum sensing to communicate with each other and coordinate their activities. This allows the microorganisms to regulate their behavior in response to changes in the biofilm environment. For example, quorum sensing enables microbes to sense local cell density so that collective functions are only switched on or off when an appropriate number of cells are present to take advantage. So a biofilm is a very complex living organism, not just of bacteria, but of many things, as we'll go through at this point, that basically make a very protective and stealthy substrate within which bacteria can freely communicate with each other, replicate and invade host cells and cause disease and make it very difficult to treat them. So biofilms can also contribute to chronic infections by promoting antibiotic tolerance among the residents. The extracellular matrix of the biofilm can provide a physical barrier that limits the diffusion of some antibiotics and other antimicrobial agents, making it difficult for them to reach the bacterial cells within the biofilm. Additionally, bacteria within the biofilm can adapt to the presence of antibiotics by activating genes whose products confer resistance to these drugs. At a more fundamental level, when residing within a biofilm, microbes adopt a profile of gene regulation that is significantly different from that of their planktonic counterparts, and one that triggers a multitude of adaptive responses that are associated with increased antimicrobial tolerance.
We had a question. Somebody wanted to know, you had mentioned Botox earlier. What do you think of the pharmaceutical Botox?
Well, when I asked Dr. Pete about it several years ago, he says there are studies showing that when people use Botox topically on their skin and it paralyzes the nerves on their face, for example, to get rid of wrinkles, it's actually transmitting a paralyzing effect in the brain, and he was highly opposed to the use of Botox. It's, injecting something that's paralyzing your nerve that then stops the muscle from contracting, and thus you have less wrinkles, but the long-term effects are not well known, and studies are showing that it actually is causing some paralysis in the brain. So I do not recommend Botox based upon that information. Wrinkles are naturally a part of aging. Yes, it'd be nice not to age, but this happens to all of us.
When formed, biofilms can trigger chronic inflammatory responses in the host, leading to tissue damage and impaired healing. The biofilm matrix can stimulate host immune cells to release inflammatory mediators, which can cause chronic inflammation and tissue damage. So currently, more than 80% of bacterial infections, this is quote unquote, 80% of bacterial infections are caused by the formation of bacterial biofilms. The most common microorganisms for medical device-associated infections are Staphylococcus aureus and Staphylococcus epidermidis. Multi-resistant gram-negative bacteria, that is E. coli, Klebsiella pneumoniae, and Pseudomonas aeruginosa, as well as Acinetobacter, Baumannii. So these organisms are well associated with medical device-associated infections. So E. coli then, which is a coliform bacterium of the genus Escherichia, commonly found in the lower intestine of warm-blooded organisms. Most strains are harmless commensals, but certain subtypes are pathogenic, causing serious food poisoning. And then briefly, Klebsiella pneumoniae, although found in the normal flora of the mouth, skin, and intestines, can cause destructive changes to human and animal lungs if aspirated, specifically to the alveoli, resulting in a bloody brownish or yellow-colored jelly-like sputum. So a lot of these organisms that we have as commensals, some of which can be introduced into other areas of the same body, can cause disease. And this again, this is the terrain, the theory of terrain by Duraford and Lapraz did a paper on this in 83, I think 83 or 84, showing that the terroir, that the French call terroir, which means terrain, gives grapes. They weren't doing studies on grapes. I was just going to go on to say that grapes inherently have a characteristic flavor that is resulting from the terroir in which they're grown. The same with microorganisms. So we can have E. coli up our nose, but if for whatever reason that same bacteria gets introduced into our intestines or our bloodstream, into our heart or whatever, can cause serious damage. So organisms, when they're supposed to be where they are commensal, they are non-pathogenic. But whenever they get into a different body part or different tissue, they can be completely destructive. So when stimulated by the harsh environment, the exopolysaccharides, fibrins, and lipoproteins secreted by bacteria adhere to the surface of inert objects and form microbial substances. Now, EPS, which is the extracellular polymeric substance, is conducive to bacterial adhesion and promotes the formation of a biofilm matrix composed of extracellular polysaccharides, exogenous DNA, proteins, and lipids. The EPS matrix reduces the effects of antibiotics by neutralizing antimicrobial agents or limiting diffusion using extracellular polysaccharides. The EPS facilitates intercellular communication, it protects cells from chemical damage, it provides oxygen diffusion, it releases extracellular enzymes for nutrition, and in turn stimulates the spread of bacteria within the biofilm. The most common pathogenic bacteria for urinary tract infections in adult women is E. coli. Now, uropathogenic E. coli, or UPEC, these strains may evade the body's immune response by stimulating a pro-inflammatory reaction or obscuring the immunogenic bacterial component. Bacterial biofilms may play a key role in maintaining the sustainability of UPEC strains in the bladder and vagina. The second most common pathogen of urinary tract infection is Proteus. When they first adhere to surfaces, Proteus mirabilis can proliferate, develop and form biofilms, enhancing the antimicrobial properties of bacterial microbes and protecting the bacteria from the body's immune function. Bacterial biofilms are an essential factor in the persistence and existence of bacterial infections, and their diversity can influence the viability and survival of bacterial microorganisms. Okay, we have another caller on the air. So caller, you're on the air. Where are you from? Where's your question?
I'm from Southern Humboldt, and you once said that you thought stevia is toxic, and I'd like you to explain that, please.
I think the leaf is non-toxic, but the products that they make, the sweeteners, the sweetener products, the process is toxic. And I don't think we actually said it was toxic, but Dr. Pete was saying that the process is the
industrial process.
Yeah, the industrial process of extracting that. It's like, when they make corn syrup from corn, you can eat fresh corn, but when you make corn syrup from corn, it's actually a really chemical process. And the same with fructose that you can buy in the health food store or in stores. The process to manufacture fructose is also quite toxic. But no, I like the leaf, the stevia leaf, the herb. It's fine to use if you like the flavor and you need a little sweetener. Does that answer your question? She's off there. Okay, I hope that answers your question.
Okay. The number, if you're in the area or out of the area, is 707-923-3911. And we can squeeze one more in, perhaps, if you want to call in between now and about 5-2, but it is 7:52. So I think what I want to say is there's plenty of different strategies for overcoming biofilms in different parts of the body. And those chemicals have got quite a bit of research done on how they get there, how they work. And definitely some of those conditions that we've talked about very recently, like SIBO and SIFO and Candida, and other infections that would be typically normally treatable quite easily, can be very resistant because of the presence of biofilms. And it's really not been around for too long that kind of biofilm busting compounds and different products have been available. But I think as time goes on, more research is being done, and previously untreatable or very resistant to treatment illnesses are being resolved with the addition of a biofilm buster.
So I just wanted to mention some other non-device related bacterial biofilm infections include dental plaque.
Right, exactly, yeah. So the plaque on your teeth is a classic biofilm.
So it's, there's a biofilm that's formed there, and your body can't kill the bacteria in your mouth because the bugs are hiding in the biofilm. Mm-hmm. So dental plaque, that's considered a biofilm. A urinary tract infection, so people that get chronic recurrent urinary tract infections. There are several causes of that. It could be like misdiagnosed as a herpes infection. cystic fibrosis, people who have cystic fibrosis, the cystic fibrosis, you form a very sticky secretions that are like biofilms and hide the bacteria, and they get chronic chest infections. Otitis media, ear infections, infective endocarditis, tonsillitis, periodontitis, so even periodontal disease and inflammation of the gums. Necrotizing, it's hiding in there where you can't reach it. Necrotizing fasciitis, osteomyelitis, infectious kidney stones, and chronic inflammatory diseases in general.
I had read ages ago that the mechanism of action of cranberry and blueberry too for urinary tract infection was that the proanthocyanidins actually inhibited the fibrils from attaching to form that biofilm. Right. So perhaps those fruits would be good for other biofilm related conditions.
Right.
Yeah.
Well, that was, we'll do it next month for sure because we don't have time at this point, but I got a whole bunch of different natural products, or the compounds within natural products, from ajoine, allicin, carvacrol, in thyme, through to
carvacrol's and oregano too.
Imodin, in Rheum palmatum, the same in Cascara sagrada, so aloimodine, vitexin from vitex species, so we'll break into that next month. Hyperforin from Hypericum perforatum, we'll
St. John's wort.
We're also looking at
and the nattokinase from natto.
Right.
Easy to make at home.
Yep, and there's plenty of citrus species that have isoliminic acid. We've got ginger, obviously, from zingerone and how it's used as a biofilm buster. Then curcumin and curcuma. Then we've talked about epigallocatechin 3-gallate from green tea. There's ginkgolytic acid, tannic acid, etc., etc. Chelorythrine from chelidonium, which is an excellent antiviral for wart treatment. But let's wrap up the show so we have plenty of time. So next month, folks, we're just going to finish up on the subject of biofilm, and then we'll get into those compounds that have definitely been used as adjunctive therapies in the treatment of some of those diseases that we've listed that have been resistant to treatment because of biofilms. So you've been listening to Ask Your Herb, Dr. KMED Garberville 91.1 FM, and we'll be back on the air again in May, third Friday of the month, from 7 till 8. So we'll carry on discussion about those different compounds that are used to break down the biofilm, so that those particular natural compounds that we've spent lots of time talking about as directly antibiotic for that particular condition may have not been as successful as they could have been had you have known about a biofilm hiding these particular species from the compound we've been disclosing as treatments for it. So until the third Friday of next month, my name's Andrew Murray.
My name's Sarah Johannesson Murray. Thank you so much for listening. We hope you have a great weekend.
Our website is westernbotanicalmedicine.com. We can be reached by email, best thing, and we also consult with clients about a wide range of conditions. So if you want to take advantage of that, just drop me an email. Thanks so much. Good night.